Okay guys, today I'm going to show you how the HIV virus replicates inside of your cells. It spreads to other cells. A lot of different processes at play. There's a couple of key terms that are critical. Okay, first, we know this. We got something called a CD4 receptor. CD4 receptor. Now that's the receptor that's on the outside of T helper cells. Okay. And that's the receptor that the HIV virus bonds to. So you have a CD4 receptor, you have a concept called fusion. Okay. You have a enzyme called reverse transcriptase. You have an enzyme called protease. And then you have an enzyme called integrase. Now, whenever you see these ACE words, ASE, that means you're talking about enzymes, okay? And enzymes are nothing more than amino acid chains or polypeptide chains or proteins, all the same thing. Okay, these are enzymes. Now the HIV virus is very unique, and it's very sinister in the way that it attacks the body, because what it does, just like all viruses, okay, it's nothing more than RNA inside of a protein coat. Well, some viruses have DNA, but this particular virus has RNA and a protein coat. Now, the interesting thing about HIV virus is that the protein coat holds 2 strands of RNA. Now, the 2 strands of RNA is because they both have unique jobs. Okay, so let's draw a cell. Keep all these key terms in mind. So first you have, so let's just draw a rough cell. Okay, let's put a nucleus, some ribosomes. Throw a mitochondria in there. Okay, so the nucleus, as we know, is what holds the genome. So you have your 23 pairs of chromosomes from mommy and daddy. Okay, so that HIV virus is going to first come into contact with the cell. It's going to bind with the CD4 receptors. These are the receptors that are on T cells. Okay, so the HIV virus bonds with the CD4 receptor and then gains access to the cell. Okay, how does this happen? The receptors that are on the viral coat unlock the keys to the receptors on the cell membrane. Okay, that happens on the CD4 receptor. So what's going to happen now is the virus itself is going to open up, and then it's going to fuse with the membrane of the T cell. Okay, and when that happens, it's going to open up the virus and it's going to dump all the viral content. When it dumps that viral content, it's not just the RNA. Okay, so it's like a toolkit. The HIV protein coat holds a toolkit of all the necessary equipment for the virus to take over the cell. Okay, so that includes reverse transcriptase enzyme, the protease enzyme, and the integrase enzyme. They all have a very important job for manufacturing the virus. Okay, so it's going to come into the cell. First thing that's going to happen, remember, the HIV is an RNA virus. That RNA is going to turn into DNA. This is a process called reverse transcription, and the name is fitting because reverse transcription, it's very similar to normal transcription. The only difference is that RNA is getting turned into DNA, and that's happening because of free nucleotides, okay, inside of the cell. They find the RNA, they make their match, boom, they make a strand of DNA. Okay, this is with the help. This can only happen with the reverse transcriptase enzyme. Okay, so that's what facilitates this transfer from RNA to DNA. So we'll just call this RT for now. Okay, so next step, enzyme called integrase is going to take both of these strands of RNA that are now DNA, okay? Think about it like this. So this is integrase. This is still happening outside of the nucleus. It's going to take that DNA, okay? It's going to tie it in a little bow, okay? So right there's the integrase enzyme, ties it up like a little bow, and then takes that DNA that's now in a little loop, pulls it into the nucleus, okay? And what it does now, so assume that this is the DNA, your DNA, not the viral DNA, okay? So this is just one little corner of a chromosome inside of your genome. And then what's going to happen is it's going to get sliced open, cut open by the integrase enzyme, and then it's actually going to insert, so it's going to pull that virus in, break up your DNA, and then insert itself into the actual DNA. And this is the really sinister thing about the virus, because this establishes lifelong infection, okay? I mean, it's a part of you. It's part of your genome, you know, just like all of the genes that you inherited from your parents are. So the DNA kind of tricked your body into thinking that it was your own DNA material, okay? Inserted itself into your genome, and now it's going to do what DNA does. It's going to transcribe and translate itself. It's going to make proteins out of the DNA, which is now the HIV DNA has a permanent place inside of your genome, and your body won't be able to differentiate between the two. So normal transcription is going to occur, okay? And this is when it's going to start manufacturing all the necessary components of the virus, because it's literally in that code, okay? So when that happens, the immature virus, the components of the immature virus are going to rise to the edge of the cell, okay? They're going to kind of coalesce. That protein code is going to get formed. Those enzymes are all been established because of the protein synthesis that happened during translation. It's all going to rise up to the top of the cell. And then right as the virus is leaving, okay, it's almost a mature virus now. You have both strands of RNA that came and kind of coalesced. And then you have this kind of jumbled up viral machinery that's inside of the cell, all the enzymes required to infect new cells and do the exact same thing that happened during entry. But what's going to happen now is it's all kind of jumbled up in one soup, okay? And the proteins or polypeptide chains are still all connected to one another. So you need this final enzyme protease, and that's going to kind of organize all that viral material, organize that toolbox, so it can become a fully functioning, mature HIV virion is what we call it, ready to infect new cells, and it does. So it has the CD4 outside, it's got the 2 strands of RNA, it has all the enzymes required to go and infect a new cell. So it happens because all 3 of these. Reverse transcriptase establishes DNA from RNA, integrates that new DNA into the genome, and then the virus is manufactured, and all of its components are as well. And then protease organizes and cuts all the peptide chains inside of the virion itself, and makes it a fully mature virus to infect other cells. Okay, this is HIV, guys. Thanks.